Okay, today we're going to be looking at the brain and the spinal cord. When we talk about the brain and the spinal cord, what we are referring to there is what we call the CNS, the central neural system. The brain and the spinal cord is the CNS, central neural system. Anything outside of the brain and spinal cord, meaning any peripheral extension of that, is called the PNS, the peripheral neural system. The PNS, peripheral neural system, meaning anything outside of the brain, meaning cranial nerves, and you need to be very comfortable with the 12 pairs of cranial nerves by their name, Roman numeral, and their function. So cranial nerves are part of the peripheral neural system, and anything off the spinal cord, which are spinal nerves, that too would be considered peripheral neural system. The only thing that is central neural system is the brain and everything within it, the spinal cord and everything within it. The neural system is an extremely important system. It develops at around day 18 or so. After sperm and egg come together, it is the neural system that is the very first system that develops. And it's not called the neural system at that point. It's actually referred to as the primitive streak, which becomes the notochord, and the notochord embryologically starts to evolve and develop into what we refer to it today as the spinal cord. So it is the neural system that is the very first thing that develops within the human body, whereas most students think it's the heart because they look for a beating heart when it comes to a living baby. But what develops before that beating heart is the spinal cord and the brain, which is the CNS, the central neural system. And the function of the neural system is really a processor, right? It ingathers and takes in information. It processes that information and then spits out a motor command. So taking in information is what we refer to as sensory, and then it processes that information, and then it sends out a command or a message, which is what we call motor. So sensory is coming in, up the spinal cord to the brain, and then motor is the messages that the brain sends down the spinal cord and out to the nerves, to muscles and organs. Sensory information ascends, meaning it goes up, and motor information descends, which means it goes down. Another term that we use interchangeably for sensory information is afferent information, afferent, A-F-F-E-R-E-N-T. And the other term for motor information is efferent, E-F-F-E-R-E-N-T, efferent information. So afferent goes up, efferent goes down. So when you hear about an afferent neuron, that's a sensory neuron. When you hear of an efferent neuron, that is a motor neuron, messages going out. In the brain, we have centers in the brain that receive sensory information. And then we have a part of the brain that initiates the output of motor information. So there's a sensory chamber, if you would, and then there's a motor chamber in the brain. And there's lots of relays within the brain as well. So meaning that sensory input can go up the spinal cord into the brain, and there is a relay or a synapse, and it says, okay, here's the pit stop, and now we're going to send this information to this part of the brain or to that part of the brain. Okay. So different parts of the brain have different functions. If you think about structure determines function, and we look here at this brain, what we're looking at is the cerebrum. And the cerebrum has different lobes. You don't want to confuse the term cerebrum with cerebellum. They sound alike, but they're different. So when you hear of frontal lobe and parietal lobe and temporal lobe and occipital lobe. Those are the main lobes of the cerebrum. The cerebellum is right down here. We see down here the cerebellum. The frontal lobe is really interesting. The frontal lobe is involved from an emotional perspective with your motivation, with your higher executive order thinking. Frontal lobe. The frontal lobe is also responsible for initiating motor activities to your muscles. But it's your driver, it's your ambition. So when you have an individual that's a go, 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 very motivated type of individual, they have a very healthy frontal lobe. When you have someone that is very comfortable sitting on the couch, lacks motivation, won't get out of the basement, is not a career-oriented individual that can adapt well to society, these are people that have frontal lobes that are not as highly activated or as functional as other people. When you hear of a person that does road rage and has anger issues, right? Someone accidentally cut someone off. Maybe they just didn't see him in the side mirror. And then that one person thinks that that person cut them off intentionally and was out to hurt them with the intent of hurting them. These are people that have frontal lobe issues. Many criminals have frontal lobe issues. Many criminals that commit murder and commit these crimes, these are frontal lobe issues. When people have the lack of ability to think, if I do this, there's a consequence for that action, they don't think that far ahead. These are people that have frontal lobe issues. Okay, so it tells you the significance of that frontal lobe. Your parietal lobe over here, your parietal lobe is more involved with your perception of things. These are people that have a lot of body image disorders, have parietal lobe issues. Hey, doc, look at my left ankle. It looks fatter and bigger than my right ankle. And I look at their ankle, and to me, to my brain, they look the same. Now, one of us has to be right and one of us has to be wrong. So either my parietal lobe is distorted and I'm perceiving balance where there's imbalance, or my patient's parietal lobe is off and they're seeing imbalance because the brain is imbalanced when there truly is balanced girth of their ankles. And this is a true story. So what I'm sharing with you is true. So when that patient said to me, doc, look at my left ankle, it's fat, it's swollen, it's so much bigger than my right. And I said, it looks the same to me, but I don't want them to feel that, I'm intentionally saying that they're wrong or it's all in your head, right? We don't want to come across abrasive. So I use objective data, objective measurements. So I said, can we use a tape measure and measure? And if it is larger, it will indicate that to us. Would you agree? They'll say, yes, yes, doc. So I take a measure and I go around the left ankle and I'll say, what number do you see? Doc, it looks like 14. Okay, let me take the same tape measure. Let's go around the exact same anatomical region on the right ankle. Can you tell me what number it is? 14. So you had 14 on the left, 14 on the right. Would you agree that if they're the same number, that they have to be the same size? And you see the confused look, and they say, yes, but they don't leave it alone. But look, can't you see that it's much swollen on the left and the right? And there's no rationalizing someone's perception of it. So these are parietal lobe issues. The occipital lobe is your visual center. We think your eyes do the seeing, but they don't. Your eyes have receptors. That's all the eyes have are receptors, visual receptors, color receptors, black and white receptors. And then it takes that information and it brings it back to the occipital lobe that allows you to see. You could do nothing to the eyes, take a baseball bat, crack someone in the back of the head, or someone slips and falls, bangs the back of their head on concrete, and they can go blind. Not because the eyes were damaged, but because the occipital lobe was damaged. Your temporal lobe deals a lot with taste and a lot with hearing. That's your temporal lobe. Here, the red, this is part of the frontal lobe. The blue strip here, the blue strip is part of the parietal lobe. This red strip is called pre-central gyrus. The blue strip is called post-central gyrus. And the separation between both is called a central sulcus. So again, you have a pre-central gyrus and you have a post-central gyrus. Pre means before, post means after. Pre-central gyrus, post-central gyrus, and the separation between the two is the central sulcus. Why am I pointing this out to you? Because earlier I said that there is a part of the brain that receives sensory information, and then there's a part of the brain that sends out motor information. So the post-central gyrus is where all of your sensory data ascends up to. And the pre-central gyrus is where all of your motor information starts, that it sends it out from. So they're really important parts of the brain. Okay, and then here is your cerebellum, which deals with balance, your sense of balance, precision and smoothness of your movement, where if I were to touch my finger to my nose and I can do it smoothly without any type of tremoring or shaking, that is a good cerebellum. If I want to throw a baseball to Rachel and I see where Rachel is and I throw the baseball to her, but I use too much power and I generate too much force and it goes over her head. My cerebellum says, all right, I'm going to make an adjustment to that. The frontal lobe initiates the movement, but the cerebellum refines it. It gets like a photocopy message and refines what you want to do to make the modification. That's what the cerebellum does. If I were to close my eyes and I do this, that is a healthy cerebellum. But if I go like this and I'm trying to touch my nose and I miss, that is a problematic cerebellum. And this is why when there are sobriety tests, when a police officer pulls someone over because they're driving out of lane and they're swerving their car and they think maybe they're under the influence, they'll pull them over and they'll do the test, touch your finger to your nose. Now, if they can't, and they smell alcohol, they're assuming that alcohol got into the brain, especially the cerebellum, and they can't touch finger to nose, which is faulty cerebellar function. Or they'll have them walk heel to toe in a straight line, which again challenges the cerebellum, which is all balance. Okay. All right, let's go back now to the beginning here. And I can scroll down and show you some of these other landmarks. So here, when we look at the cerebrum, which is the main part of the brain, I can see a left cerebral hemisphere and a right cerebral hemisphere, right? This is the front and this is the back. So we have two hemispheres, a left cerebral hemisphere and a right cerebral hemisphere. Both hemispheres create different functions, where your left hemisphere is more your logic, your linear thinking, your science, your language, your mathematics. That's all left side of the brain, whereas your right side is more creative, more artistic, more music, more art, more facial recognition, right? Babies have a really strong right hemisphere of the brain because they have to be able to look at their mommy or daddy and recognize their face. They also have to recognize if their mommy or daddy is upset with them or angry. So they study the eye movements. They study how big the eyes are. They look if they're smiling and there's teeth, or they get scared because they think they're angry. So right and left brain is very important. They look different, slightly different. They have different functions. These um, hills, right? There's hills and valleys that you can see around the brain. The hills are called gyri or gyrus, and the valleys are called, or the grooves are called sulcus. Now we're looking at the brain from the front, right? You could see the clear skull here. So this is the left cerebral hemisphere. This is the right cerebral hemisphere. And again, you see the word cerebral, which refers to cerebrum. It's not cerebellum. There's a difference. If I'm talking about the cerebellum, you'll hear me say left cerebellar hemisphere, right cerebellar hemisphere. They sound alike. Don't be fooled. Don't be tricked. When we say left and right cerebral hemisphere, it refers to the cerebrum. The outside, the outer part of the brain is called the cerebral cortex. The term cortex means bark. I don't mean like a dog barks. I mean the bark of a tree. It's the protective covering that goes around a tree. That's what the cortex is. The brain has gray matter on the outside, and it has white matter on the inside. What is gray matter? What is white matter? White matter is white due to his appearance based on myelin or the myelination. When you do neurology in lecture and you look at the different type of nerve cells, there's one main cell called a neuron, which is electrically excitable, and then there are other supporting cells that are called neuroglial cells or neuroglial cells, meaning nerve glue. And two of the cells, one is called a Schwann cell, the other is called an oligodendrocyte. Both of those produce myelin in the nervous system, except one myelinates in the central neural system, whereas the other myelinates in the peripheral neural system. So the one that myelinates in the central neural system is called the oligodendrocyte. The one that myelinates in the peripheral neural system is called a Schwann cell. Schwann cell. Myelin is needed to speed up nerve conduction. It's the protective layer of fat and cholesterol that wraps around neural tissue. It's an insulator and it speeds up the impulses of conduction of those action potentials. If the body is incapable of producing myelin, or the body creates antibodies against the myelin production, that is a disease that we call MS, which is multiple sclerosis. And typically the first place in the body where the signs and symptoms show is the eyes. Okay. So here is a dissection of the brain, and we can see the outer layer is the cortex and the inner layer is white matter. When we look at the brain again, we'll see the left and right cerebral hemispheres, and there is this fissure that separates the two. It's a long fissure, so we call it the longitudinal fissure. It's in alignment with that sagittal suture of the brain. I mean, the sagittal suture of the skull. So that's called the longitudinal fissure. Now, the brain, we think we can separate into a right and left half. You really can't, not without slicing it. It looks like there's a slight separation here, which is true. But if you put your fingers in a real brain, as I have, and you've tried to separate them, there are neurological fibers that connect the left brain to the right brain, and you literally have to slice it with a knife to separate the two hemispheres of the brain. Here in the skull, we can see these blue, looks like these blue vessels. And when you do arteries and veins in anatomy and physiology too, and you do the cardiovascular system, you'll be able to go over these landmarks again. These blue structures are part of the venous system that return blood and is responsible for dumping and draining blood from the brain back into the heart. Okay, so this is the superior sagittal sinus. Below it is an inferior sagittal sinus. Here is the straight sagittal sinus, and then here is the transverse sagittal sinus. You'll see those again in anatomy and physiology too. Here we're looking at the brain from the posterior. So back here, this is the occipital lobe that's green. Remember, that's where your visual center is. Here is your cerebellum, and separating the cerebrum from the cerebellum is another fissure called the transverse fissure. This is the longitudinal fissure. This is the transverse fissure. This is your left cerebellar hemisphere. This is your right cerebellar hemisphere. And this is the vermis between the two. Vermis means verm, which is worm in German. This layer that surrounds this translucent layer right here is called the tentorium cerebelli, especially right here. This is where the cerebellum will sit. And this is going to separate the cerebellum from the cerebrum. It's called the tentorium, like a tent, a tentorium cerebelli with two L's referring to cerebellum. Remember, here is the longitudinal fissure. This fissure going across this way is the central sulcus. It's not a fissure, it's a sulcus. This central sulcus is significant because if you can identify the central sulcus in front of it, right, this is your frontal lobe, back here is the occipital lobe. So once you identify the central sulcus, in front of it is the precentral gyrus. Behind it in blue is the post-central gyrus. So central sulcus, precentral gyrus, post-central gyrus. What's the function of the precentral gyrus? That's where your motor output begins. What's the significance of the post-central gyrus? That's where all your sensory data ends up. And if you were to look up the term homunculus in your textbook or under, let's say, Google Images or something, and you look at the homunculus, you're going to see this very, very odd form and shape of a human being stretched completely over both the precentral and post-central gyrus. And it stretched over the pre and post-central gyrus in a fashion based on the complexity of movement or the amount of sensory input that it perceives. So your fingers do a lot of perception, sensory to feel, but you can also control precise movement of your finger. As you can movement of your eyes, you can really create smooth, controlled movement, which means in terms of brain space that it will take up, it's going to take up a lot of brain power and brain space. But large muscles, like let's say your quadriceps and your hamstrings, they're not going to take up a lot of brain power and brain space, although they're large and immense structures. They move more grotesquely, lacking precision. So the things that require more precision and control need a lot more neurons to control it. So it's going to take up more space. That's both afferent and efferently for sensory or motor. So here, again, from the side view, this is the lateral view. Here's your frontal lobe. Here's your occipital lobe. Here's your temporal lobe. There's your central sulcus. In front of it, precentral gyrus. Behind it, postcentral gyrus. Separating the parietal lobe from the temporal lobe is the lateral sulcus here. If we take the brain and we turn it upside down, these yellow structures, these are cranial nerves. So that's part of the peripheral neural system. So here is the olfactory tract, and here's the olfactory bulb. This is really part of all cranial nerve number 1, which is the olfactory nerve, which controls the sense of smell. Here and here, this is your optic nerves. Optic nerve is cranial nerve number 2, and that controls vision. Where both of the optic nerves crisscross, they make an X here and here. That point right at the center is called a chiasm, the optic chiasm. And there is a structure right in between called the pituitary gland. The pituitary gland, if there's a tumor there and it gets larger and it expands, it can actually put pressure on the optic nerves, creating visual disturbances or different blindness that can take place in the eye. So you want to make sure that you're very comfortable and familiar with the 12 cranial nerves, their names, and their function. That will help you not just with lab, it will also help you with lecture. Okay, here is what we had already gone over. There's your frontal lobe, here's your parietal lobe, occipital lobe, and temporal lobe. You know what their primary functions are as well. Now here we have the frontal lobe. So this precentral gyrus is part of the frontal lobe, and the blue, which is the postcentral gyrus, is part of the parietal lobe. Notice it's also referred to as the primary motor area. Why? Because this area is where motor function is initiated. Okay. The area just in front of it is a motor association area, meaning they're connected. You're going to have fibers from here running to here, back and forth. So they're just an association area. But these are your two primary areas: primary motor, primary sensory. So here in the blue, you can see the parietal lobe, and this blue structure is the postcentral gyrus, aka primary sensory area. And just behind it are somatic sensory association areas. They're linked together. Here is your occipital lobe in the back, and this is the visual cortex right back here. And there's also visual association areas. So how do these work, these association areas? Your optic nerves, which pick up visual data, it's going to send it to the visual cortex. So right now, you can all see this image and you can all see me. It's the visual cortex that says, I see something. But now there are going to be signals that go from the visual cortex to the association area. And you could then say, oh, that's a model of the brain. Or, oh, I see that person, that's Dr. Lindner, right? So the visual primary cortex and the association areas are working together. Here is the temporal lobe, and this is where your primary auditory area is, and below it is the auditory association area. So you may hear a song on the radio, or iTunes or something like that, right? So you can hear music playing, that's the auditory cortex. But then underneath it, you're going, I wonder which artist that is. Do I recognize that voice? Right? That's the auditory association area. When we dissect and move the temporal lobe down, we see that there's this orange area referred to as the insula. It's just deep within the brain. When we cut the brain in half sagittally, right, down the longitudinal fissure, what we can see here is a corpus callosum. And what the corpus callosum is these fibers that connect the left and right hemisphere together. And they're sending information to left and right hemisphere, right? They don't function independently. They are communicating to each other. So it's the corpus callosum that are these neuron tracks or these axons that go from left hemisphere to the right. And what they've discovered is that women have a stronger corpus callosum and more fiber connections in the corpus callosum than men. So women have a stronger crosstalk between left and right cerebral hemispheres, which may be why women may be better at multitasking than men. It's just in the wiring. Okay, then we have the septum pellucidum, which is this clear translucent membrane. And sorry about that. And here, the septum pellucidum serves as a membrane that's involved with your ventricles. Okay, the ventricles, we'll go over shortly, is where cerebral spinal fluid flows. And we have 4 ventricles. We have 4 ventricles. Then we have an area here that's called the fornix. So you have corpus callosum, septum pellucidum, fornix. There are a few other structures down below, which we'll go over in another slide. Again, just on another model, here's the corpus callosum. Here's the septum pellucidum, and you see it says lateral ventricle because that's going to separate your 2 lateral ventricles. One lateral ventricle is here in the right hemisphere, and then there's a mirror image of it of the same thing in the left cerebral hemisphere. And then inferior to that is an area called the fornix. This is your cerebrum. Here is your cerebellum. This is your medulla. And this is your pons. And right here is your midbrain. I'll make a little box here for it so you can see me outlining it. So you have medulla at the bottom, pons, and then right here is the midbrain, right in this area. Medulla, pons, midbrain. Cerebrum, cerebellum, and the diencephalon is everything here in the center. Diencephalon actually means through the brain, so it's in the center of the brain. We'll go through some of those landmarks. Here we're looking at the brain underneath. You could see the olfactory bulb and the olfactory tract here. This indentation is where your lateral ventricles, right? It's the cavity for the lateral ventricles. And I'll show you what the shape of that, and then you'll understand why the cavities look this way. And the septum pellucidum is just the membrane that separates the lateral ventricles from one another. So here's the corpus callosum. Here's the septum pellucidum, or the lateral ventricle. Here's the fornix. Right here, this is the thalamus. This round, large structure is the thalamus, and the little yellow, looks like a nucleus there, this little focal area is called the interthalamic adhesion. The purpose of the thalamus is a major, major relay station for all of your sensory input. So every bit of sensory input that comes in to the brain is relayed first to the thalamus, and then the thalamus will relay it up to the post-central gyrus. Okay, all of the sensory input goes to the thalamus first, with one exception, the exception of the sense of smell. The sense of smell does not go to the thalamus. So again, corpus callosum, septum pellucidum, fornix, thalamus with the interthalamic adhesion. Below the thalamus is the hypothalamus, right? The term hypo means below. It's the hypothalamus. the function of the hypothalamus controls the autonomic neural system. What is the autonomic neural system? It's your sympathetic and your parasympathetic. Sympathetic is fight or flight response. Your parasympathetic is feed and breed, or rest and digest. It's more of your relaxation portion. Sympathetic speeds things up, like your heart rate and your breathing, whereas the parasympathetic slows those things down. The only thing that's reversed is digestion. So the parasympathetics, when it's stimulated, it speeds up digestion. When the sympathetics are stimulated, it slows down digestion, which is why you're not supposed to be in a fight or flight response and then eating a meal. You'll have indigestion. You're supposed to eat when you're parasympathetically stimulated. Okay, let's take a look. Here is your thalamus. There's your interthalamic adhesion. Below the thalamus is the hypothalamus, which would be here. And then the hypothalamus connects to the pituitary gland right here. There's connections from the hypothalamus to the pituitary gland by way of the infundibulum. This is the infundibulum. The pituitary in AMP2, you will learn the pituitary has two divisions, an anterior and a posterior pituitary. The pituitary is sometimes called, especially the anterior pituitary, is called the adenohypophysis, and the posterior pituitary is called the neurohypophysis. There's the term hypophysis. You may remember that this was called the hypophyseal fossa. The hypophyseal fossa is where the sella turcica is, that Turkish saddle. So you have hypothalamus, infundibulum, pituitary gland. Sella turcica, aka hypophyseal fossa. Here is the choroid plexus. This is a important structure in terms of producing cerebral spinal fluid. It's called a choroid plexus. It's a capillary system here. And on the posterior side, you could see the pineal gland. It's pretty small, but it's right there. That's the pineal, aka pineal gland. The pineal gland is involved in secreting melatonin. The pineal produces melatonin, which helps to tone you down at night so you can sleep. That's the pineal gland. We'll take a closer look. Here's the choroid plexus. Here's your thalamus, the interthalamic adhesion, the hypothalamus. Here's the pineal gland. Again, the pineal produces, don't confuse pineal with pituitary. They're very, very different. Here are three other main parts of the brain. From inferior to superior, it's the medulla, the pons, and the midbrain. Medulla, pons, and the midbrain. There's a few cranial nerves that originate in the medulla, and that would be 12, 11, 10, 9, and 8, or 8 through 12 originate in the medulla. 5, 6, and 7 originate in the pons. 3 and 4 originate in the midbrain. And 1 and 2 originate in the cerebrum. So if we start at the bottom, it's 8, 9, 10, 11, and 12 in the medulla, 8 through 12. In the pons, it's 5 through 7. In the midbrain, cranial nerve 3 and 4. And in the cerebrum, it's cranial nerve 1 and 2. And they're numbered in order that they're actually developed embryologically. So the first one to develop is cranial nerve 1, which is the olfactory. The second one that develops is cranial nerve 2, which is the optic. Cranial nerve 3 is the oculomotor. Cranial nerve 4 is the trochlear. Cranial nerve 5 is the trigeminal nerve. Cranial nerve 6 is the abducens. Cranial nerve 7 is the facial nerve. Cranial nerve 8 is the vestibulocochlear nerve. Vestibulocochlear. Cranial nerve 9, glossopharyngeal nerve. Glossopharyngeal. Cranial nerve 10, the vagal nerve or vagus nerve. Cranial nerve 11, spinal accessory nerve. Spinal accessory. And cranial nerve 12 is the hypoglossal. And you should know them by name and by Roman numeral, as well as their functions. So we can determine, right, especially if there's head trauma or a type of concussion, what part of the brain is affected by testing cranial nerves. So if they're hearing buzzing in the ear or they're having hearing problems, I would say, okay, well, that's the vestibulocochlear, cranial nerve 8. That means the problem is in the medulla, because that's where cranial nerve, the nucleus for cranial nerve 8 originates. If someone's having problems with eye movements in a particular way, I would have to say, all right, is it cranial nerve 3 or cranial nerve 4? Because those two originate in the midbrain. Maybe there's a problem or a concussion that's affecting the midbrain. If someone's having trouble with smell, then the problem is up in the cerebrum. Okay, so once you know how to test the cranial nerves based on what their functions are, if you know what they do, all you have to do is find a way to test it. If there's decreased function, then we know there's something wrong with the structure, because structure determines function. So that's a pretty significant or relevant way of relating cranial nerves to everyday function and if there's any type of concussion. Okay, just a close-up view of the medulla, the pons, and the midbrain. Okay, these two structures are part of the midbrain. Now, keep in mind that there's two of these, like they look like two bumps or hills. Two of them are on the right hemisphere, and then two of them are on the left. So there's really 4 bumps, and I'll show you what those 4 bumps look like in a moment. Here they are. So here in this picture, we're looking at just half of the brain. So these are the 2 bumps. When we look at both hemispheres put together, here is the medulla, here is the pons, and here is the midbrain. And the midbrain has 2 bumps, 1 on each side on the top and 2 on the bottom. So when you count these up, we have this what's called body of 4, or corpora quadrigemina, which means body of 4. We have 2 superior colliculi and 2 inferior colliculi. The superior colliculi is where the input of visual information goes to. And then we have auditory information, which will end up synapsing and being relayed to the inferior colliculus. So when your eyes are tracking and moving and following something, you do need the superior colliculus to work. When there's a loud bang or a noise coming off to the side and you hear it, and then your head turns and you protect yourself due to a loud noise, we know that the inferior colliculus is working. The pons has a lot to do with controlling the rate and rhythm of your breathing patterns. That's your pons. The medulla is super important because that's really Houston control. That's where cranial nerve 8, 9, 10, 11, and 12 originate, with cranial nerve 10 being the biggie, which is the vagal nerve. And the vagal nerve has motor and sensory connections to every major part of the human body. The cardiac center and respiratory control center also lie in the medulla, which is why if there's any type of brain trauma at the medulla, the heart can stop and breathing can stop. The medulla is protected by the top 2 vertebrae in your neck called C1 and C2. C1 and C2, C1 is the atlas, C2 is the axis. So you can have subluxation or slight structural malalignment or distortion of C1 or C2 that can put pressure and interfere with the transmission of neurological input or output to the medulla, creating dysfunction. We see this a lot in infants when they're born. Sometimes there's birth trauma where there's too much force used on an infant's neck or spine. When they pull the baby out of the vaginal canal, sometimes the doctors use too much rotational force. And if there's too much rotation of the head, it subluxates the top C1 vertebrae. That could put pressure on the medulla, distorting the vagal nerve, throwing off digestive function. So when babies are breastfed or they're tried to be bottle fed, they just projectile vomit. They can't hold down the food because the valves going into the stomach aren't getting the signals, so they never open and the baby just throws it out. These are babies that really need to get checked at that C1, C2 level, get that area adjusted by a chiropractor that knows how to make those corrections to help the baby flourish, for sure. Okay, here is the same structure. This is posterior view, right? Up here is the brainstem from the posterior. Now we take the same thing and we rotate it around so we could see it from the anterior view. You don't see the corpora quadrigemina, but you could see those big optic nerve and the optic chiasm there. You could see the medulla, pons, and the midbrain. Pons means bridge because it connects to many parts of the neural system. These bumps here are called the pyramids, the medullary pyramids. Here is the cerebellum, the right cerebellar hemisphere, the left cerebellar hemisphere, and right in between we see the vermis. Again, labeled right and left cerebellar hemispheres, and you see the vermis. Don't confuse cerebral hemispheres with cerebellar. Cerebellar with the two L's refers to the cerebellum. And here's the vermis in between. When we separate both hemispheres, right and left cerebellar hemispheres, we can see that we have the difference between white matter and gray matter. So here is white matter that looks like the veins of a leaf. That's called the arbor vitae, which really means tree of life. And then the cortex out here is also known as folia, F-O-L-I-A. Folia means foliage. It looks like a leaf. So the white matter is the veins of the leaf. That's called arbor vitae. And then the cerebellar cortex is the gray matter, which is referred to as folia, F-O-L-I-A. Now the next few structures is here, these are the ventricles. And when we look at the ventricles, you will see that we have lateral ventricles 1 and 2, lateral ventricle 1 and 2. This is what sits in the cavity. This sits in the cavity of that cerebrum, right? So you have 2 ventricles, a left and right ventricle, also known as ventricle 1 and 2. And then here's the third ventricle. In order for the cerebral spinal fluid to go from the lateral ventricles into the third ventricle, it has to go through the interventricular foramen, also known as the interventricular foramen of Monroe. It goes from lateral ventricle 1 and 2. It's got to go through the interventricular foramen, and then it ends up into the third ventricle. Remember I said there's 4 ventricles. So here's the third ventricle. From the side view, here's the lateral ventricle, here's the third ventricle, and here's the fourth ventricle. For cerebral spinal fluid to go from the third ventricle into the fourth ventricle, it has to go through another drainage pipe called the cerebral aqueduct. So you have Lateral ventricle 1, lateral ventricle 2. It goes through the foramen of Monroe to get into the third ventricle. The cerebral spinal fluid goes from the third ventricle through the cerebral aqueduct to make it into the fourth ventricle. That's the flow of cerebral spinal fluid. It's produced by these ependymal cells, and these ependymal cells are going to line the choroid plexus. Okay, that's where cerebral spinal fluid is produced. Then, within the fourth ventricle, how does the cerebral spinal fluid exit? Well, it's got to go through the lateral apertures. These are lateral. And then it also goes through the median aperture. So you have lateral apertures and median apertures. That's how the cerebral spinal fluid exits. Okay. Okay, that was the last slide that's there. Let me stop this for a second, and then we'll start the next. Let me stop the share and the record.